Mr. Majewski's Anatomy 32 class lecture, Chapter 20, Part 1, Somatic Senses. So, it turns out the word sensation has a very specific definition, and this is the conscious or subconscious awareness of change. So basically, to get a sensation, first you need some kind of stimulus, a change in the environment. In the picture below, the change in the environment is the finger touching a sharp needle on a cactus. Then you need some sort of sensory receptor, something in the finger that is going to detect this change in the environment and then signal a neuron to produce a nerve impulse that will then go to the central nervous system. So then you need the sensory neuron itself, which conducts the signal to the brain, and finally the brain needs to receive and integrate this information, deciding on an appropriate response to this sensation. So sensations can be classified in two really big categories. You can have general senses, for instance the somatic senses of tactile sense, thermal sense, pain, and proprioceptive sense, which are found throughout the entire body. Pretty much if someone touches you anywhere on your body, you should be able to detect that. Then another category of general senses are the visceral senses. As we talked about in Chapter 19, they're pressure, stretch, chemicals, nausea, hunger, and temperature, all involved with the autonomic nervous system. Then the other big category of sensations are the special senses. The special senses are ones that are located in the head, and they include smell, hearing, taste, vision, and equilibrium. However, for this chapter, we'll be focusing on the somatic senses. So, you have to have some sort of somatic sensory receptor that is going to detect the change, and then a somatic sensory neuron that's going to carry the signal to the central nervous system. Sometimes the somatic sensory receptor and the somatic sensory neuron are actually the same cell. So, sensory receptors can be categorized in a number of different ways. One way to do this is by the structure of the receptor. One big category are free nerve endings. So here are some examples of free nerve endings found in the sample of skin here. These free nerve endings are the dendrites of the neuron that are receiving the signal. Another category are encapsulated nerve endings. Here again you have the dendrites of the neuron that is in this case wrapped around by layers of connective tissue. And it's these dendrites that then send the signal through that same neuron to then take it to the central nervous system. And then the third way is by a separate cell that acts as the receptor that then passes the signal on to the sensory neuron. Another way to categorize sensory receptors are by the type of stimulus detected. So it could be many things: photoreceptors in the eyes, mechanoreceptors, say in stretch in a visceral organ, thermoreceptors for body temperature, osmoreceptors and chemoreceptors detecting changes in the blood composition, and nociceptors, which detect sensations that lead to the feel of pain. Third way to categorize sensory receptors are by the location. You have exteroceptors, which are located on the outside of the body and are detecting changes in the environment outside of the body. Interoceptors, usually found in visceral organs, are the inside the body, and they detect changes to the organs and structures inside the body. And then there's proprioceptors, which are found in skeletal muscles, and they detect changes in stretch in joints and in muscles that could be caused by your own active decisions or by some sort of external environmental factor, like getting hit with a baseball. So now we're going to talk about tactile sensations. These are senses that are detected through our skin. Now first, the most common tactile sensation is the sensation of touch. And this gets broken down into two big categories: fine touch and crude touch. With fine touch, you're detecting very specific types of touch. You'll be able to tell exactly where that touch is occurring, whether it's, say, rough or smooth, things like that. And the fine touch sensation is primarily detected by two types of receptors. You have the type 1 cutaneous mechanoreceptors, formerly known as Merkel discs, as seen here, which are saucer-shaped flattened free nerve endings. And the type 1 cutaneous mechanoreceptors are called slowly adapting. This means that they detect the sense of touch, and they keep detecting it. So as long as you're touching that thing, these receptors are saying, yes, you're touching it, you're still touching it, you're still touching it. And this is different from the corpuscles of touch, as seen here, also known as Meissner corpuscles, which are a mass of dendrites inside an egg-shaped capsule. And Meissner corpuscles, or corpuscles of touch, are rapidly adapting. So they say, yes, you've been touched, and then they stop. And they don't send another signal until something changes in what you're touching. Crude touch, on the other hand, are perceived by their own two types of receptors. You got the type 2 cutaneous mechanoreceptors, as seen here. These are elongated, encapsulated nerve endings again, and they are slowly adapting. So they will say, yes, something is touching you. They can't give you a lot of information except the general area, but you know you're being touched. Versus the hair root plexus, which are seen here, where again you have three nerve endings, in this case wrapped around the hair follicles. And when something causes that hair to move, say a breeze or a mosquito landing on your leg, you get a rapid signal saying, yes, something's touching you here, and then it stops. Another kind of tactile sensation is that of pressure and of vibration. These two are closely linked. So pressure is basically defined as deep tissue deformation, and it can be detected by the corpuscles of touch, the type 1 mechanoreceptor, and the new one here, the lamellated corpuscle, also known as the Pacinian corpuscle. And pressure basically means the difference between something pushing on you gently versus pushing on you really hard, being able to distinguish between those two types of pressure. And the lamellated corpuscle, as seen down here, is an oval structure with many, many layers of connective tissue wrapped around dendrites of a sensory neuron. And they tend to be found deep in the skin between the dermis and the subcutaneous layer. Now, vibration is another kind of sense, and this is detected by both the corpuscles of touch and the laminated corpuscle. And basically, if you think of vibration as being constantly touched and then not touched back again and again and again, this allows you to feel that vibration effect. For instance, if you ever went roller skating and then you stopped roller skating and you still kind of felt like you were on the roller skates, that's an echo effect of the tactile sensation of vibration. Two other types of tactile sensations are itch and tickle. Itch and tickle are both thought to be detected by free nerve endings of specific sensory neurons. Itch is usually stimulated by certain chemicals, so if your skin gets slightly damaged or if a certain chemical ends up on your skin, then it'll stimulate these free nerve endings, causing the itch sensation. Tickle, on the other hand, is a little less well understood. It's thought to arise from free nerve endings, and basically the reason why they think you can't tickle yourself is because the cerebellum is able to detect that you yourself are doing the tickling, and therefore it is not something you need to be aware of, and so you aren't. Another type of tactile sensations are thermal sensations. These are detecting temperature through the skin. Again, this is also caused by free nerve endings, so the sensory receptor is the same as the sensory motor neurons. You have two different kinds of thermoreceptors. You have cold receptors, which detect from 50 degrees Fahrenheit to 105, and warm receptors that detect from 90 degrees Fahrenheit to about 118 degrees Fahrenheit. When you detect thermo temperatures that are below or above this range, then you instead trigger the pain receptors, letting you know that this is too hot or too cold. And then there's the nociceptors. Pain. Pain is a good sensation. You want to know when something is hurting you. That way you stop letting it hurt you. Nociceptors are also thought to be free nerve endings, and they are found in every tissue in the body except the brain. The brain doesn't feel pain. And they tend to be stimulated by chemicals released from injured tissue or by any stimulus of sufficient intensity. So for instance, if you are walking around in the dark and you turn on the lights real quick, you feel pain because the amount of light coming in is just way too much for your retina to handle. And there are two types of pain. There's fast pain, which is acute, occurs in less than a second. It's like touching that needle, it's ouch, and then it's done. And then there's slow pain. Slow pain tends to last for seconds or more. It can last days, weeks. It can be really bad. And it can be very excruciating. Now, pain sensations are localized in different ways. Fast pain tends to be very localized at that point of the damage. Slow pain, on the other hand, especially if it's pain in your visceral organs, can feel more spread out and actually can be kind of distant from where those organs are. So, for example, that pain in your lungs and diaphragm could actually localize up to your neck and shoulder area. Or pain to your kidney could actually be anywhere on your waist and even on the side of your legs. So it can be difficult to tell from where you feel a pain exactly what it is that is wrong with you. Proprioceptive sensation. This sensation is basically letting you know where your head and limbs are located and how they are moving. And that's why these sensorizations are localized to inside the skeletal muscle tissue or within the tendons themselves. Also, it can allow for weight discrimination. So you can pick something up that weighs a pound, try to pick something up weighing 10 pounds, you can tell the difference in the weight. So there are two types of proprioceptors. There's the muscle spindles that are found within the skeletal muscles. They consist of several slowly adapting nerve endings wrapped around 3 to 10 intrafusal muscle fibers, specialized muscle fibers just for this sensation. And their function is to measure the muscle length, how much stretch is occurring in the muscle, and when you should be really worried about it. They have alpha and gamma motor neurons. The alpha motor neurons are responding to signals from the sensory axons of the muscle spindle to contract the fiber. So the alpha ones are saying, hey, you need to contract. And the gamma motor neurons, on the other hand, are just adjusting the tension of the muscle spindle itself so that it can detect when your muscles are even further stretched. The second, proprioceptors are found in tendons. They're called tendon organs. They help provide information to protect the tendon and muscle from damage due to excess muscle tension. So again, they are detecting how much stretch is going on, trying to let you know when you're doing too much stretching. Also, there are a number of other receptors found in joints, and they're called the joint kinesthetic receptors. They include things such as the type 2 subcutaneous mechanoreceptors, the tendon organ, laminated corpuscles, free nerve endings. Basically, they're there around the synovial joints to try to help you know when those joints are feeling pressure, if they're accelerating or decelerating, if there's excessive strain. Again, basically lets you know where your body parts are and know when those body parts are being pushed beyond their limits. Finally, there's this disorder called phantom limb sensation. And this is where an amputee experiences sensations associated with where that missing limb used to be. And while it's not completely understood why this occurs, one interesting way to treat it is for the person to use a mirror and basically stimulate the limb they still have while looking at the mirror so that it seems like they're stimulating the missing limb. And sometimes for some people this relieves the sensation they're experiencing, especially if that sensation is itch or tickle or something like that. And that's it for this part of the lecture.